Before we introduce our speakers, I would like our president to just say a word of welcome. Tama, over to you.
Thank you very much, Fanas, and good afternoon, everyone. Greetings from winter Cape Town. It's good to see you and to be in your presence this afternoon as we take the second SASSB webinar. I mean, particularly, I'd like to thank Fanas and the FABI team for continuing to give us the privilege to get together as a community to share our research and to keep in touch. And this is our second webinar. Well, first one having been on AI, looking at big issues on diseases and global change. And today's webinar moving more close to my heart, especially, and for us in Africa, where malaria and other parasites are important. I'll tell you, as a kid, I'm embarrassed to say every time I got malaria, I told my mother that I was not sick. Not because I was not unwell, but because I feared the injection. The days of the reusable needles, which were as long as a needle used to inject cows, which would be put down your butt and you cannot be able to sit for a couple of days. I'm glad that research is going on this, and I'm hoping that as especially climate change forces us to deal with the parasites, extending out of the range, that will not have to go back to those needles again. On the SSB side, I would like to just inform you and remind you of our forthcoming conference, where we hope to meet you in person in Stellenbosch from the 5th to 7th of July. This is a meeting co-hosted with the International Polychaete Societies conference, and we're looking forward to being able to come together as a community after months of years of disruption by COVID-19. Thank you again, and we look forward to it. Good to see you. Fanas, back to you and Kelsey to take us the next steps of the meeting.
And over to Kelsey. She will be our chair today. And as we've said, we will start off with the South African speakers, and then when hopefully by that time Susan has joined us and we can have the main talk. But we're looking forward to all the talks. Thank you very much. Over to you, Kelsey. Sorry, you muted.
Thanks, Fanas. It's been a while for me. So our first speaker this afternoon is Dr. Shüné Oliver. She is a researcher at the Wits Research Institute for Malaria, and has been at Wits researching the Anopheles mosquitoes. And she's here today to tell us about anthropogenic pollution as a driver of evolution of insecticide resistance in Anopheles. So thanks, Shüné.
No problem, Kelsey. Let's first just start sharing, and I would like to apologize for not having video, but my camera doesn't work very well. So let's just get this going. Share. I'm assuming that everybody can see my screen. Yeah, I think so.
Yes.
Okay, let's put it into presenter mode. Excellent. Okay, so good afternoon, everyone. As Kelsey has said, my name is Shüné, and I am split over the National Institute for Communicable Diseases and the Wits Research Institute for Malaria. And I'm going to ease you into the session with a fairly simple talk. And unfortunately for you, I'm fairly certain it's going to be a talk about something that you are not going to be particularly sympathetic about. Suffice it to say, it is doubtful whether mosquitoes top the list of anyone's favorite animal. In fact, if I had a rand for every time I was asked the question, why do we need mosquitoes? And what exactly would happen if we get rid of all the mosquitoes? I'm fairly certain I could fund all my research activities on my own. So let's just get that question. Out of the way. What is the third? Why is this an issue? The problem is most people think of mosquitoes as this monolithic entity, just this thing that we can deal with, rather than a complex group of species that differ vastly in all aspects of their biology and behavior. So I must say that the general hatred for my beasties is probably for a good reason. Mosquitoes are by far and away the most dangerous animals in the world, being responsible for more deaths than any other organism on the planet. So how is it possible that these small flies can just be so deadly? Now, despite the fact that ticks harbor a greater variety of arbovirus and parasites, there's simply no comparison when it comes to the mortality burden that is induced by mosquitoes. A range of pathogens have co-evolved with mosquitoes, but by far the most significant of these happens to be malaria. So let's take a look at the animals responsible for transmitting this disease. Now, for those of you who are not familiar with entomology, it is important to state that not all mosquitoes transmit malaria. This is an important fact. Anopheles mosquitoes are the only species that transmit malaria to humans. And of the 3,500 known species of mosquitoes, 140 Anopheles mosquitoes can be found in Africa. And of those, only around 40 species are capable of transmitting malaria. It is worth noting that the diversity of Anopheles species is greater in Africa, but it's like in Asia, but Africa happens to be home to the most efficient vectors of malaria in the world. Mosquitoes and humans, as well as the pathogens that they transmit, have been co-evolving together for a very long time. It is believed that the first species to adapt to biting humans is Anopheles fenestus, which happened to change their behavior and began associating with humans as soon as permanent settlements became the norm with the advent of agriculture. And to this day, Anopheles fenestus is still the most efficient vector of malaria in the world. Human activity has had a major effect on mosquito behavior, as well as driving insecticide resistance. And these are a few of the aspects I will be covering today. A classic example of the effect of humans on mosquitoes is the case of Culex pipiens in London. Culex pipiens is known as the common northern house mosquito. So if you imagine the noisy, busy thing that irritates you at nights, in South Africa, it will probably be Culex fasciatus. In the northern latitudes, it's probably going to be this mosquito. What happened in London, however, was that a subpopulation decided to go underground, quite literally. This was very unusual behavior, but a portion of their population began moving into the Underground Railroad network. And yet, within about 100 years, we now have two separate species, which are reproductively isolated. Furthermore, the above-ground species, Culex pipiens, has distinctly different behaviors from the below-ground species, Culex pipiens molestus. This includes variation in mating behavior, biting preference, and the activity in general. This rapid evolution has demonstrated how critical an effect us humans can have on mosquitoes. Besides affecting the behavior of mosquitoes, probably the most significant anthropogenic recent effect that we've had on the mosquito is insecticide resistance. As we can see from this map over here, looking at the dots in red, where they're indicating insecticide resistance, this problem is growing, and it is growing at a rapid rate and increasing with time. The issue is that insecticide resistance is more than a matter of the mosquito just being able to survive a lethal dose of insecticide. It has a range of pleiotropic effects and largely affects almost every aspect of the mosquito's biology. It is important to note that this selection can drive mosquito evolution, as different mechanisms can be selected that results in the same resistance phenotype. Now, for those of you who are not familiar with mosquitoes, may think that what I'm showing you here is simply a collection of mosquitoes, pictures of the same mosquito. That is not what it is. These are members of a complex, a group of morphologically identical related species that differ in their biology and behavior. As I said earlier, what Africa lacks in comparative Anopheles diversity compared to Asia, we make up for in the sheer efficacy of our vectors. We have some of the most efficient vectors of malaria in the world, such as Anopheles fenestus, which is the most efficient vector of malaria in the world, but also, importantly, we have members of the Anopheles gambi complex. And if we look at this figure over here, these are the members of the Anopheles gambi complex. And you will see that they differ in their behavior, as in where they bite and where they rest, and how well they transmit malaria. However, I'm not going to be talking about all these mosquitoes. I'm going to be talking about one specific mosquito. This magnificent lady over here is Anopheles arabiensis. Anopheles arabiensis is one of the most adaptable mosquitoes you are likely to meet, because this mosquito will do whatever it needs to do to survive. It will bite humans, it will bite animals. It's not particularly fussy about what it eats. It will bite indoors and outdoors. And it is this outdoor activity that results in it maintaining a low level of outdoor transmitted malaria known as residual malaria, which is difficult to deal with because you can put in place every vector control effort that you want to, but if the mosquitoes are biting outdoors, it is going to be a problem. And this is the case with Anopheles arabiensis because they are difficult to control because they primarily rest outdoors. And the problem with that is that all of our most efficient vector control tools happen to target indoor resting mosquitoes. This is things like indoor residual spraying, insecticide treated nets. As you can see, these are all going to be things that are going to be effective if the mosquito is active inside. But when the mosquito is active outside, it's a lot more difficult to control. The reason why I'm interested in this mosquito, besides the fact that it's just a generally interesting mosquito, is that it is the major malaria vector that we find in South Africa. And it is important to note that not so much in South Africa, but in the rest of Africa, they are adapting to breeding in urban areas. So they are spreading the range in which they do their business. And this means that malaria is no longer just a disease of the rural areas. So why is this happening? This has simply got to do with pollution and their adaptation to pollution. Now, members of the Anopheles gambi complex very traditionally like to breed in clean, clear, sunlit bodies of waters. And this was one of the partial reasons why it was restricted to where it was restricted. However, members of the complex, specifically members of the complex that like to bite humans, are adapting to breeding in polluted water. And this is particularly true for Anopheles arabiensis, which is doing well and learning to thrive in polluted waters, and this is facilitating its move into urban areas. Now, the work that my group has done has focused a lot on understanding the effect of pollution on this member of the Anopheles gambi complex, Anopheles arabiensis. What we have done is to test the effects of different pollutants ranging in toxicity, so metals are highly toxic, whereas fecal pollution is not that toxic. And we have looked at the differential effects that this has on insecticide-resistant and insecticide-susceptible Anopheles arabiensis. So if we start with the most toxic one that we tested, so the heavy metals, when we looked at resistant mosquitoes, we found that there wasn't a huge amount of developmental effects, and it didn't affect their longevity at all. In fact, what we found was that these mosquitoes actually had an increased insecticide tolerance, and the only real downside to this was that it did reduce their fertility, particularly from the male side. Now, herbicides are not as toxic as metals, but again, here we have the resistant mosquitoes having the advantage. Being bred in herbicide water gave them a developmental advantage. They developed faster. Their adult longevity was increased, and again, they had increased insecticide tolerance. When we started looking at less toxic pollutants, such as inorganic fertilizer, we see that here the susceptible mosquitoes start having an advantage. They had the greatest developmental advantage, and susceptible mosquitoes developed faster in the fertilizer water than the resistant mosquitoes. Neither of them had an effect on longevity, but again, when it comes to the resistant mosquitoes, again, breeding in fertilizer water gave them increased insecticide tolerance. And finally, when we looked at fecal pollution using organic fertilizer as a proxy, we see that insecticide-susceptible strains, as well as species, because we found this not only in Anopheles arabiensis, but other members of the Anopheles gambi complex, they had the greatest advantage. Particularly in terms of the time to the larval development time, how long the adults lived, how big they were, And this was the first time that we saw an increase in insecticide tolerance for both insecticide susceptible and insecticide resistant mosquitoes. But again, when it came to increased insecticide tolerance, the advantage went to the resistant mosquitoes. Crucially, we have found that a single larval exposure to pollution can affect the next generation, even if the next generation is not exposed to pollution. If we take a quick look at this graph over here, this is essentially a measure of tolerance to insecticide. And here we can see this is their baseline tolerance to the insecticide. And after a single metal exposure, their tolerance increased significantly. And although if you breed the second generation in polluted water, you'll get an amplification of this effect. If you breed a second generation in clean water, that effect does not disappear, and they still have an increased tolerance to pollution. And what we found later was this was due to epigenetic modification within the first generation. Another factor I am interested in is the insect microbiome. And this is because commensal microbiota plays a critical role in the life history of all insects. The gut microbiome in particular plays an important role. And it is quite critical to understand the biology of this commensal microbiota to provide insights in key life history traits, particularly for mosquitoes. And the traits I'm most interested in are toxin degradation, because that is going to affect their capacity for insecticide resistance, as well as their natural defense system, because the immune system of the mosquito determines whether they transmit malaria or not. Now, what we knew from our previous studies was that larval environment altered the gut microbiome, because we had an osmotolerant member of the Anopheles gambi complex, known as Anopheles mirus, and we found that the concentration of salt water that these mosquitoes were reared in altered the gut microbiome diversity. And crucially, we found that adults emerging from high salt concentration water had greater bacterial richness and more antiplasmodium protective bacterial genera present than those that were grown in fresh water. So therefore, our next question was, could larval exposure to a toxic pollutant have a similar effect? And to a degree, the answer is yes. What we clearly found was that larval exposure to cadmium and copper altered both alpha diversity, which is the within species diversity, and the bacterial beta diversity, the between species diversity. The significance of these changes is that the larval exposure to metals reduced gut bacterial diversity in wild KwaZulu-Natal Anopheles arabiensis. And although it is known that metal exposure can drive insecticide resistance, it may have a different effect on the capacity of the mosquitoes to transmit malaria. This is because the bacterial genera most overexpressed in these metal treatments happen to be protective against Plasmodium. And this was true for copper for those that were treated with copper, as well as cadmium. Over and above the potential to modulate critical life history traits, such as the capacity to transmit Plasmodium, the larval environment is also an important consideration for vector control, not only when it comes to driving insecticide resistance, as we have discussed, but also for an intervention known as paratransgenesis. This is the term for a form of biological control that indirectly genetically modifies an organism through the use of bacteria. The bacteria you can use can either be genetically modified, or you can use protective native symbiotic bacteria. As we know that larval exposure to heavy metals alters the microbiome, it is important to consider the larval environment if we are to embark in any form of paratransgenesis interventions for vector control. Now, I have just given you a taster of some of the effects of human interaction on mosquitoes. So what should your take-home message be? The first thing that you have to understand is that human activity has very clearly shaped mosquito behavior and biology. The selection for insecticide resistance has affected a range of life history traits and is currently driving evolution of the same species in different geographies. Insecticide resistance has pleiotropic effects on mosquito life history, and that insecticide resistance affects the larval response to larval pollutant exposure. Larval exposure to pollutants can have transgenerational effects, which appears to be mediated by epigenetic alteration, and larval exposure to pollutants can alter the gut microbiome. This has the potential to alter vector competence, because from what we see, metal exposure appears to select for Plasmodium protective bacterial genera in South African Anopheles arabiensis. However, it is important to know that we still do not understand the effects of other pollutants on the gut microbiome, and this needs to be examined. So this is important to bear in mind because we need to understand whether pollution can affect paratransgenesis as a vector control intervention in South Africa. So in summary, if you forget everything else that I've said, just know that human activity is selecting for insecticide-resistant mosquitoes, and this is particularly true for larval pollutant exposure on the very adaptable malaria vector, Anopheles arabiensis. The current evidence suggests that this effect will persist for a few generations even if we reduce or remove the source of pollution. But what this means for malaria transmission still needs to be determined because we do not have a conclusive answer as to whether insecticide-resistant mosquitoes make for better vectors. So at present, although we know that this effect is happening, we cannot say whether our activities happen to be selecting for more efficient transmitters of malaria. Over and above my funders that I would like to thank, this work would not have been possible without my group of dedicated students, both past and present. Each of the students here have contributed in part to some of the data that I have shown. So as I've said, I am very grateful for them, and this presentation would not have been possible without them. And crucially, I would also like to thank Dr. Givmo Mwenhenga and his SIT team for providing me with wild mosquitoes, which I would otherwise not have been able to have access to. And thank you to everybody for paying attention to my very brief overview of what we are doing to mosquitoes. Thank you very much. Escape.
Thanks so much, Shüné. That was really interesting.
Okay.
I have a question, and if folks in the audience have questions, please feel free to either raise your hand or type it into the chat. But I was wondering about pools, like how,
Yes.
if as mosquitoes are dispersing into urban areas, so I guess it's a two-pronged question. How are they getting here? And are they going to use pools? And what will that do?
What we have, Zach, when you're talking about pools, normally chlorine is a particular problem. And as yet, we have not found any species of mosquitoes, whether malaria transmitting or not, breeding in heavily chlorinated water. What we do find is if you find mosquitoes breeding in your little child blow-up pools, those tend to be mosquitoes like Culicidae longreolata, which happens to be mosquitoes that bite dogs and birds. So Anopheles mosquitoes are a bit on the back foot. They have developed they are adapting slower than other mosquitoes. So our Culex mosquitoes and Aedes have adapted to breeding in a range of polluted water, including sewage. And it must be noted that you do find common house mosquitoes breeding in areas with sewage. So I can only imagine that it is a matter of time that the Anopheles the Anopheles adapt to it as well. How they are getting there, I believe is a twofold, there's potentially a twofold answer to this. And the one is movement of people from rural areas to urban areas, bringing mosquitoes along with them. And the second issue has got to do with potentially with climate, and that as areas that were potentially too cool for mosquitoes becoming warmer and warmer and warmer for a longer period, it is making it more amenable for mosquitoes to move in. So again, this is another effect that we are having on the mosquitoes, and that the general idea is that it seems to be that we are moving the mosquitoes along from their natural habitat, from the rural habitats, and then making the urban environment more available to them. But in terms of pools specifically, I haven't checked the chlorine yet, but there has not been any reports of mosquitoes breeding in heavily chlorinated water.
Okay, that's good news, particularly for urban areas.
Yes.
Great. Thanks, Shüné. If there are no other questions, we will have more time for questions at the end. So if you think of any, hold on to them. And I'd like to introduce our second speaker, Dr. Mamohale Chaisi, who is a research scientist at SANBI in the zoological research of foundational biodiversity science. And so, Mamohale, if you're able to share your screen. There we go. Great. Thank you so much.
Sorry. Yeah, thanks again for the introduction and for the invitation. So it is a pleasure to present our talk or our research here in SANBI on avian malaria. So just to briefly show my face, that is me, but I'll turn off the camera. So unlike in humans, in birds, malaria is actually caused by species of 3 distinct genera. And this include Plasmodium, Haemoproteus, and Leukocytozoon. And these actually cause malaria and related diseases in birds. So malaria is actually insect transmitted, and it is transmitted by different insects again, and different species of mosquitoes in this case. So there's really much more diversity than in humans. So the severity of infection depends on the parasite lineage, host, age, and the immune status of the host. And avian malaria and related diseases are among the most pathogenic. These parasites are actually amongst the most pathogenic species of wild birds, with reports of declines of native bird populations globally. And they therefore negatively affect host fitness and reproduction. So their host range and geographical distribution is expected to increase due to global climate change. And this, of course, is a phenomenon for all vector-borne diseases. So it's recognized as a significant threat to the conservation of animals and of birds in particular in this case, as well as to food security. And penguins and other seabirds are highly susceptible, and infections are actually, like I indicated, of significant conservation importance. And they also affect domestic birds and therefore do impact on food security. Free-ranging birds, including migratory birds, usually act as reservoir for infections to captive birds. And the life cycle, as I indicated over there, is a typical cycle of Plasmodium with alternation of both asexual and sexual stages in the vector and in the host. So those are actually the infective stages to the mosquito, or as I indicated there, and there is quite a huge diversity in terms of morphospecies. So these are intracellular parasites found in red blood cells mainly, but they actually differ a lot. Identification is quite tricky and requires a trained eye. So they are highly diverse, and over 250 morphospecies have so far been described, but genetic diversity is actually indicates a presence of close to 5,000 lineages. And these are cytochrome B haplotypes which have actually been developed globally. And there is actually a dedicated database for this, where this information was obtained. So there is currently a limited reports on the occurrence and effects of these parasites on birds generally, especially in South Africa. We don't really have a lot of reports on the effects of these parasites, either in captive or free-ranging birds. So there's a need for increased surveillance in birds to determine their impact on the health of birds, especially in threatened avian taxa, where they can actually contribute to the decline of species. So the aim of the study was to carry out a molecular survey of avian haemosporidia in selected populations of captive and free-ranging birds in South Africa in order to determine different aspects of their biology. And this is just a summary of the samples that we analyzed. So we analyzed just over 600 samples originating from different species of birds. And I've indicated those that are actually threatened, but we also analyzed samples from free-ranging birds, and also included mosquitoes that were actually collected from around the penguin enclosure here at the Pretoria Zoo. And some of these birds, especially from penguins and from the crown hornbills, are actually post-mortem cases. So these were suspected clinical cases of avian malaria. And most of these samples are actually stored in our huge collection of biomaterial at the SANBI Biobank. So we analyzed this using standard molecular-based techniques and used different tools for sequence and phylogenetic analysis. We also analyzed the samples using quantitative PCR in order to quantify infections. And we compared the nested or conventional PCR results with those from qPCR. And what this just indicates is that in most cases, the qPCR actually detected far more infections than nested PCR. And generally, qPCR is regarded as more sensitive than nested PCR, so it wasn't really much of a surprise. And in wild birds, which are actually represented by this first group here, you can see that infections were really high. They were actually over 80%. So looking at the lineage diversity, so this are actually the lineages that we identified from the first study that we did on Afrotropical birds. And so the samples actually originated from South Africa and from West Africa, parts of in Uganda and Nigeria. So the lineages that are highlighted are unique lineages that we actually identified or were reported for the first time from our study. And I've just indicated the lineage. This is actually Parahemoproteus lineage that was actually the most commonly identified. But then we identified one sequence actually from one of the cuckoos, and which was quite distinct from all the other lineages. And this is actually a leucocytosome lineage, and I'll refer to this as I proceed. So looking at the phylogenetic relationships, we managed to obtain about 155 cytochrome B sequences from the samples that we analyzed. And this just shows the distribution and of the lineages or the evolutionary relationships that were actually observed. So again, from all these sequences, 2 of these were actually unique haplotypes. 43 of them are described for the first time from the study. And these actually represent possibly 22 distinct species, as I actually highlighted with those red dots. And this is mainly based on their percentage identity and genetic distances. So although in most cases, we really did not do any morphological studies because the slides were either absent or were not of very good quality. But in some cases, it has actually been reported, in some studies, it has actually been reported that genetic distances of over 5% actually represent distinct morphospecies. However, it is still essential now to properly characterize these all this diversity that was observed. So this is just the distribution of the species, putative species that we actually obtained. And what is actually of interest is this particular species, Haemosporidia antigonis, which is exclusively a parasite of cranes. And even in the studies, we only identified it from cranes. So it is actually regarded as Haemosporidia species, but as can be seen here, it is quite distantly related to the other Haemosporidia species. So the next slide just indicates host parasite relationships. So usually in some cases, these lineages can actually be host specific. They infect certain birds only, but in most cases, they actually are not host specific and can actually be isolated from a variety of hosts. So this is just the distribution of the hosts that we actually observed. So the first one was actually mainly from the Afrotropical birds. So this is the wild birds. And the second group is also from wild passerines. And these are actually from captive owls. And this was, yeah, a unique lineage, again, that was only identified from owls. And as can be seen, the next one was identified both from the African penguin as well as from the wild birds. And the next one was exclusively from birds. And this last group here actually is the lineage which we actually identified from post-mortem cases of penguins. So this is actually the lineage that is we actually regard as, responsible for the death of some of our penguins here in the zoo collection. And moving on to Haemosporidia antigonis, as I indicated, this is exclusively in the literature is actually described. It has so far only been described from cranes. So we actually identified it from all three South African crane species. But just to note that there are actually very few studies that actually report this particular species. And most of the reports are actually from threatened crane species in North and South America. But from the literature, again, there are these two lineages that I've highlighted that are actually described from the East African gray-crowned crane. And as can be seen, we actually identified similar sequences from the cranes in South Africa. So these can actually be regarded as African lineages. So for cranes, again, we determined the host distribution because in this case, at least we had samples from different parts of the country. And the keys over here just indicates particular species. So we actually identified the first group, the blue cranes, the second group, the gray-crowned crane, and then the wattled crane, which is the last group. So in fact, the last group is actually of mixed host infections. We actually had very few samples from wattled cranes. So as can be seen, I think in this particular center, it's actually a captive facility for cranes in the Eastern Cape, had the most diversity. And again, unfortunately, because these samples were actually not collected for this particular samples, we mainly used samples that were already in our biobank. So we couldn't really define these, and yeah, there were no blood smears. But we do plan, if possible, to actually go back to this facility and do proper sample collection so that we are actually able to describe, these species in full. And then moving on to leukocytosome, again, we do see the same pattern. In this case, leukocytosome is actually infects more hosts, or there's actually less host specificity in this case than with Haemoproteus and Plasmodium. And the lineage that I actually referred to as previously, where we only identified it from one bed, it seems to be a very common lineage now in South Africa. And from subsequent studies, we actually identified it from a whole range of beds, as can be seen here. And so this is the report from the study that was done from the penguins, from mosquitoes that actually collected around the enclosure of the African penguins. And in this case, again, we did identify this lineage. So this, in this case, we actually identified it from Culex mosquitoes, and these are possible vectors now for transmission. So we think that these are actually the mosquito species that are responsible for transmitting these parasites now to our captive animals. So there have been some recommendations for the revision of genus Haemoproteus. From the first slide that I indicated, actually Haemoproteus is divided into two subgenera. There's the subgenus Haemoproteus and the subgenus Haemoproteus. But you can actually see from this figure that there is actually a huge interspecific diversity between the two subgenera. So due to this reason, and because they're actually transmitted by different insect vectors, there is actually generally a suggestion that they might actually represent two distinct genera. And this is what we have actually seen in our studies as well. And then for Haemoproteus antigonis, these are the samples or the sequences that actually originate from birds, cranes in South and North America. And they also form a distinct clade there that is separate from the rest of the para-Haemoproteus group. So there's actually a high interspecific diversity between Haemoproteus and para-Haemoproteus, which actually suggests that they might represent distinct genera. And looking at Haemoproteus antigonis in particular, there's also high diversity within the group, and it also represents, as can be seen. So this is actually from our studies. The green area here represents the American sequences, while the African sequences indicated with dotted lines. So again, you can see that there is actually high intraspecific diversity and possibly the presence of two distinct, at least two distinct species in this case. And our results are actually supported by intraspecific sequence divergence, as is actually plotted in this case. So this is just an analysis of maximum intraspecific versus minimum interspecific or nearest neighbor sequence divergence. And the threshold here is the 5% threshold of species delineation, as I indicated earlier. And as can be seen, most of these actually fall beyond the 5%, which actually indicates a high level of diversity. And this is actually Haemoproteus, so it's quite diverse from its neighbor, which would actually be probably para-Haemoproteus. So in conclusion, our studies actually indicate that these parasites are quite common and diverse in birds in South Africa, and infections might pose a potential risk for the decline of bird populations. Some of these lineages have actually been associated with fatal infections in the African penguin. And this also highlights the importance of monitoring these infections in captive bird populations in order to support conservation efforts. So there's still a lot that we don't know about. We don't know what actually drives this diversity, but we know that recombination does occur both in the vector and in the host. And in some cases, these vectors actually, or these parasites actually infect non-adaptive hosts, where they can actually also mutate. And wild or free-ranging, as well as migratory birds, have also been implicated in the distribution of infections or transmission of infections, as well as alien bird species. We still don't know whether some of these parasites are actually co-invasions. They were probably actually introduced into the country together with alien bird species. And of course, the biggest issue is still with climate change that has actually resulted in the introduction of new vectors that might also carry some of these new or unique lineages. So there are also implications of this biodiversity or disease epidemiology. We still don't know again how these lineages actually affect the health of our birds. And there is also a need to describe the putative species that I have talked about. So some of the challenges are in the differential diagnosis of mixed infections. So because of this huge diversity, then you tend to actually have co-infections being common. However, the common assay that we actually use to determine infections does not differentiate between the different lineages or different species. So even sequencing of a single PCR product, as you know, will only result in one sequence. So there is actually an underestimation of parasite diversity. And there has been efforts now to actually develop more specific assays for actually differentiating between these different parasites. And we did try to use this multiplex assay, but I think because of the huge genetic diversity now with these parasites in South Africa, we couldn't really differentiate between the different species as is indicated in that first picture. So it still becomes a challenge. And we have also attempted to actually use metagenomics now to determine mixed infections and to see, how that could actually contribute now to what we have observed. But again, the recovery was quite low. And that is actually another challenge because these are intracellular parasites. So it is really a challenge to actually get, a pure isolate for next generation sequencing. Co-infections are also very common, not just within this group, but also with other parasites, as well as, so the first picture actually shows other blood parasites that are usually found with these haemosporidian parasites. And this actually complicates the disease epidemiology because it increases the severity of infections. And in most cases also compromises the diagnostic tools that are used to identify these parasites. And these are also all vector transmitted or mosquito transmitted diseases that might also occur with the haemosporidia in different beds or in different hosts. And these will also, of course, complicate the disease transmission. So this work was actually a team effort, and I would really like to thank everybody who contributed to the study, as well as to SANBI and the NRF, which provided most of the funding. Thank you.
Thanks so much, Mamohale. That was really interesting. I had no idea there was such a diversity of parasites, particularly in birds. So while others think of their questions or type it into the chat, I did have a question for you. And I was wondering, so as a non-disease ecologist, and parasites are very much outside of my realm, can you test how virulent each of the parasites are relative to each other, so that there might be, so all these parasites are present, but how would you know which one is actually making the birds ill?
Okay, I think, the first issue would actually be identifying, the infecting species. It is actually known from the literature which species are actually the most virulent. So Plasmodium relictum, for example, is actually an invasive species in most areas, and it is quite virulent, but it's also highly diverse. But with genetic tools like, and next generation sequencing, it is actually possible, to identify specific markers that are responsible for virulence. So it is possible.
Wow, that's really cool. I just noticed a hand that came up. Nicola?
Hi, yes. Thanks, Mamohale, for a fascinating talk. I also am a bit blown away by all the diversity. I was thinking about your comment about that you may actually have two different genera there. And your tree there, it looked as if your one subgenus was sister to something. Was that another genus?
Let me just share again.
It was a circle tree.
Is it this one?
Yes.
Oh, okay. Do you mean, are you referring to Haemoproteus and leukocytozoon in this case?
Yes, yes. And then also your yellow and red clades.
Anyway, the thing is, genetically, they're also quite similar. So in most cases, Haemoproteus actually does cluster, very close to leukocytozoon, although the genetic distance would still be, as sufficient to actually differentiate them. But they do cluster together. And that is why Haemoproteus is actually, in this case, regarded, this specific subgenus is regarded as, a totally separate genus now from Haemoproteus.
Okay, yeah, because it looks like if any one of those clades is a genus, then it looks like you've got a whole lot of separate genera there rather than subgenera, because if the subgenera aren't even sister lineages, it does argue for them being separate genera, or you recognizing them as separate genera, I guess. Yeah, thanks.
Okay, thank you.
Thanks. Are there any other questions? If not, I have some unfortunate news in that Susan will be unable to make it in time, so we will have to reschedule her. I suppose when the university president says jump, you ask how high. So hopefully we'll find a time that we can reschedule Susan for. But I would like to say thank you to Shüné and Mamohale for taking the time and sharing your work with us. I found it particularly fascinating to think about parasites and mosquitoes and the urban challenges and the avian diversity. So thank you very much.
Yes, thank you from my side as well. And then if there's no other issues, please remember to have a look at the advert for our conference in July in Stellenbosch. We would love to hear about parasites and bacteria and fungi and birds and plants and whatever you're working on that's alive and needs a systematic way to classify them. So please join us there as well. So we hope to be able to see you all, and we will let you know if we have been able to reschedule Susan's talk. Thank you very much.
And thanks for hosting us, Venice.
It's a pleasure.
Thank you. Thank
Have
you.
a good
Bye, everyone.